All right, welcome back. Hope you all had a good break. I had a fairly uneventful one, traveled to Ohio, watched the Michigan game twice, watched it Saturday, watched it on Sunday. It was so fun. All right, so we got 2 more weeks to go, plus finals week. We're getting down there, last blast. I did want to advertise our BMS seminar this week. We are slowly trying to get that started again after almost a year and a half of not having any seminars. So this one could be interesting and talk about some things that we've talked about in class. So if you are at all considering research as a career, if you're thinking about going to graduate school or maybe to be a PhD or get a master's degree, doing some research, it's good to, even if you really don't know what they're talking about after the first couple minutes, it's good to kind of understand how science is done. So I encourage you to go to that. It's right before this class on Friday over in Manitou, so not a long walk. And then I thought we'd start with a couple of review questions. All right, so kind of short ones here. Kind of questions that you might find on that first third of the final. So remember, I would encourage you, if you're starting to think about studying for that final, you want to especially know this most recent stuff, because I'm going to ask questions about that. Maybe go back and study cell structure or genetics, but the chances that I'm going to ask one of those questions is a lot less. So make sure you know this stuff. Okay, this is just a simple, straightforward morphology type question. So genera of gram-positive, aerobic, rod-shaped organisms. We haven't done a whole lot of organisms here for me to confuse you with a bunch of extra stuff, but the staph and the strep are cocci, right? Clostridium are mostly anaerobes, so that leaves the bacillus. Okay, so not a lot of pathogens. The one we talked about was the anthrax there, and the Clostridium was the botulism and the tetanus, and the C. diff, which we're going to finish today. Okay, one more specifically about botulism. A minute there. Right, so remember, it's the babies that get botulism by either inhaling the spores or eating the honey. Okay, so that's the babies. So how do adults get it? Typically, if we ingest spores, that maybe a couple of times has ever caused disease. If somebody was immunocompromised or didn't have a kind of a fully functional digestive system. Most often, if adults get it, and it's very rare, by ingesting the toxin. Okay, so usually maybe from some food that's been improperly stored, improperly canned, and the organism proliferates and makes the toxin, long after the toxin will persist in the can, long after the organism dies. Okay. Okay, questions. Where we left off last time, we were kind of finishing up Clostridium, I believe. So Clostridium, gram-positive, rod-shaped organisms, primarily anaerobes, often strict anaerobes. So these are diseases that are going to proliferate in places in our body that are essentially without oxygen. We just kind of mentioned C. diff here kind of briefly, didn't go into too much detail with the virulence factors. Treatment of this nasty, persistent diarrhea, first thing that they always try is vancomycin. We take that in pill form, maybe try a couple different courses of that. And when that doesn't work, oftentimes that will work, especially if somebody maybe young and otherwise healthy contracts C. diff after going on some other kind of antibiotics. A treatment, an alternative treatment that we've been doing this for a little while, and it hasn't really taken off. We're only doing about 10,000 of these a year. They abbreviate it FMT, so that stands for fecal microbiota transplantation. Okay, so remember, most people that get C. diff diarrhea, C. diff they had as part of their normal microbiota. They took some antibiotics that kind of cut down the variability of the organisms in their large intestine and allowed the C. diff to just overgrow. So how do you get back that balance of lots and lots of different organisms to keep the C. diff in check? Okay, you repopulate that with somebody else's stool sample. So fecal microbiota transplantation. When they first started doing this, they would do this with an endoscope. They would usually get a relative that was healthy and screen them and use some of their stool sample and just put it back in there. They can do this now by pill form. When they first started doing it, the only capsules that would dissolve sort of in the right place and not dissolve till they got to the large intestine were clear. So really ick factor there. Now they can put it in other capsules that it doesn't look so nasty, the stool sample, but patients still have to take about 10 of these for about 3 to 5 days. So it's a lot of pills, but it has a really high success rate. The problem is it's a procedure that's not approved yet by the FDA. It's still considered experimental. I tried to look up, when hospitals will do this. The Cleveland Clinic will do an FMT if a patient has gone on a course of vancomycin and has 2 stool samples that test positive for C. diff. Then they will go ahead and do the FMT. Again, usually they get a relative to donate. There is a company in Massachusetts that looks for people to be fecal donors. So less work than maybe being a plasma donor. They especially look for, healthy, maybe, young people, especially that haven't been on a lot of antibiotics during their lifetime. So, keep that in the back of your mind if you need some extra cash. There was a couple of patients a few years ago that died after a fecal transplant because they picked up a infection from their donor of antibiotic resistant microbes. So we need to get better at screening those samples, but it has, again, a lot of potential to be, to clear this from a lot of people that have chronic diarrhea. So. All right, any questions about C. diff, botulism, tetanus from last time? Okay, so the mycobacterium, with this material, I think I had posted those 5 slides talking about the final. So that's at the front end of this. So we're kind of making our way through, we started with gram-positive pathogens, the cocci, the bacilli shaped. We're going to move into the mycobacterium, which remember, these are our genera that's considered acid fast. They have that special cell wall that's often not considered either gram-positive or gram-negative. It's kind of its own special class. Some textbooks kind of group the mycobacterium in with the gram-positives because they don't have an outer membrane like the gram-negatives do. But they don't stain very well with that gram-positive gram stain. It's in your open stacks. It's chapter 21.2. I think chapter 21 is the respiratory pathogens. So this is the 21.2 is bacterial infections of the respiratory system. So there's a lot of different mycobacteriums. Most of them are considered biohazardous. So if you remember back early in the semester, we did acid fast stain. If you're in lab, we did have one species that was considered safe to work with, Mycobacterium smegmatis. There's a couple of really famous ones though, Mycobacterium leprae, the causative agents of leprosy. There's actually a couple different kinds of leprosy. There's a kind that just makes a person's skin kind of numb. And there's another kind that's called lepromatous leprosy that causes one to progressively lose their body parts. This is an organism that grows best in a little bit slightly lower body temperature, like 30 degrees Celsius. So it's going to grow best, fingers, nose, ears, toes, those kinds of places. And again, eventually causes those digits, for example, to fall off, and patients get other kinds of infections when that happens. So this is very treatable now. Obviously, treating at this stage of this patient, they're not going to grow their fingers back, but it's very treatable. We don't put people in leper colonies anymore because it's treatable with antibiotics, also known as Hansen's disease. After, I assume, the person that elucidated the cause of it. And then Mycobacterium bovis causes we call it bovine TB, bovine tuberculosis. So that's mostly in cattle, but it can infect any animal with hooves like deer and elk and moose. So that's also something that people can contract primarily from drinking unpasteurized cow milk. So that used to be really prevalent back before we pasteurized milk. Not at all common today. Again, it's a much milder form of human tuberculosis. We'll spend most of our time here talking about the organism Mycobacterium tuberculosis. This is TB in humans, also known as consumption. That was kind of an old-fashioned term for it, maybe, because it used to really consume patients, also known as the White Death. This has been called the most successful pathogen of mankind, right? So people have been getting TB for millennia. And in the US, we don't worry about it so much anymore. There's less than 10,000 cases of this a year in the US, but worldwide, this is an astonishing figure. 1 out of 3 people worldwide have tuberculosis. And that doesn't mean they necessarily have active infectious tuberculosis. As we'll see, it's a very complicated disease, kind of hard to diagnose sometimes, slow moving, different stages of it. But again, over 1 million people die of this per year, closer to 2 million. Especially prevalent here, sub-Saharan Africa, India, Southeast Asia. Again, so it is something that we thought would go away once we came out with the first antibiotic to treat it in 1946. It has not gone away. And just some more figures. I don't have numbers as to, before the 40s when we didn't have antibiotics. I assume it was much higher than this. but at least in recent history here, numbers have peaked during the AIDS crisis. So early 90s, TB and HIV were a bad combination. They often went together. That's primarily why sub-Saharan Africa has had a huge problem. But cases are going down. This was a figure I think I got from the CDC. Of course, the goal has always been to eliminate TB because it doesn't have animal reservoirs. It's just something that humans are passing around. So we should be able to get rid of it. We have antibiotics to treat it. You know, we're getting closer to the goal here, but it's not something that anybody projects that we're going to be able to do anytime soon. So if we look at the occurrence of this organism and how we pass it around, like I said, humans are the only reservoir. It's a little different form that we see in the Mycobacterium bovis causes a little bit different disease, although it's related. Okay, and transmission, droplet nuclei. So remember, those are the really small particles that we cough out, sneeze out, you know, come out when we're talking, that are so small that they stay suspended in the air for long periods of time. Okay, so this is considered airborne. Anybody can get TB. It's a true pathogen. There's certain people that are a little bit higher risk. Anybody that's HIV positive and does not have replication of that virus controlled by drugs. Somebody that's staying on their drug regimen perhaps would not have a higher risk. Likewise with diabetes, I'm talking about somebody that has maybe uncontrolled diabetes that is not, taking their medication to keep their blood sugar at the right level. If the level of sugars in the blood and the tissues is a little bit higher than it should be, then the pathogen just has more access to that sugar and is going to grow a lot faster. In addition, people with diabetes have decreased blood flow to their extremities, so they don't have as many white blood cells getting where they need to be to fight off the infection. And just sort of a catch-all, people living in congregate settings, you know, jails, for example, you know, cities with, you know, high population, you know, people that are living right on top of each other, they're going to be at a higher risk. You know, people that are immunocompromised, of course, or kind of have a lower standard of living, malnourished, that sort of thing. And of course, it matters like how close you are to somebody that has the TB, and how long you spent next to that person, and you know how much they're coughing out in the air, how many bacilli, that sort of thing. Okay, I will just mention a couple of virulence factors here. The biggest weapon that Mycobacterium has, of course, is their unique cell wall. Remember, that's composed a lot of kind of unique things, but the thing that we focused on was that mycolic acid. That's a long chain fatty acid. Remember, it's fatty acids that make up lipids. So you think of this as being really kind of waxy cell walls. So some special properties that gives the organism. These are organisms that actually will live quite happily within macrophages. So they often will allow themselves to be phagocytized by the macrophage, but then they don't get digested when the lysosome fuses with that phagosome, again, because they have this nice cell wall that just makes them quite impermeable to some of those enzymes in the phagolysosome. When they are coughed out in the environment, they're much more, they're going to dry out really, really slow. They are able to persist often for weeks out, maybe on some surface. They're resistant to a lot of antibiotics. Again, that cell wall is just really impermeable, a waxy cell wall. So we really have a much smaller repertoire of antibiotics that will penetrate through that cell wall. And that's why we needed a new staining procedure. So kind of, again, those of you in lab that I got to start reviewing, remember we added that carbylfusion as a primary stain that's it's dissolved in phenol, so it will penetrate through that lipid cell wall. And then cells that are not decolorized with that acid decolorizer, acid dissolved in acetone, are considered acid fast. So these are the pink cells here. It's kind of hard to see them here, but they're kind of short rods that tend to kind of clump together. Okay, anything that's not acid fast takes on the counter stain, which is blue. Question?
Do we categorize mycobacteria by catalase at all?
That's a good question. I don't know whether they're catalase positive or negative. I assume that they're catalase positive. It's something that's living in our lungs. They're going to be exposed to a lot of oxygen. But I would have to look that up. The vast majority of organisms are catalase positive. So usually if it's not something we talk about, it's probably positive. Another repercussion of having these specialized cell walls is that the organisms grow really slow. And that doesn't seem to be to their detriment, but it takes a long time to manufacture these long chain fatty acids. So they do grow slow. Infections usually progress over a number of years. When they're slow to grow, they're also slow to die. So when we go in antibiotics, people have to be on antibiotics for a long period of time, and that makes it definitely harder to treat. Other virulence factors, this glycolipid called cord factor. We don't really understand how that works, but it's in the cell wall of the organism. And we know that when we make a mutant of Mycobacterium tuberculosis that doesn't have cord factor, that the cell doesn't cause an infection. So we know that's important part of that cell wall. We know that it's important for the cells to kind of clump together. Again, we don't really understand why that's important, but it, again, maybe has some role in inhibiting the chemotaxis of neutrophils towards the cell and making it withstand those enzymes from the lysosome, or even inhibiting the lysosome from fusing with the phagosome. And there are other things that Mycobacterium can do that just make them more successful in living in that macrophage. Things like causing the phagosome to fail to acidify, or we didn't really talk very much about this, but macrophages have kind of two levels of activation, kind of a normal going around eating up trash, garbage in our body activation, and then kind of a extra burst of activity. They keep the macrophages from having that burst of activity. So it gets complicated, and there's just a lot that we don't really understand about how Mycobacterium tuberculosis is successful. Let's take a look at the infection that it causes. Tuberculosis can end up infecting many different tissues and organs in our body, but it almost always starts in the lungs. So typically a patient will inhale some bacilli, and then those will migrate to the macrophages that live in the alveolus. So alveolar macrophages deep within the lungs. The bacilli will replicate freely in those macrophages. And how does that act to the patient? They get these mild flu-like symptoms. So a little fever, kind of, achiness, headache, nothing too serious. At this point, some patients appear to be able to conquer the bacteria. They get, even though the macrophages are targeted, they get some presentation of antigen by macrophages to get B cells and T cells involved. It's an intracellular pathogen, so we need those cytotoxic T cells. And apparently, sometimes the host's adaptive immunity gets a jump start enough that the infection gets cleared. Other times, and I don't have good numbers about what how likely a host is to fight it off or not. But it appears that oftentimes our immune system isn't quite able to get rid of the organism, and the organism is not quite able to replicate freely either. So we get sort of a stalemate, and we get these tubercles formed. So a granuloma is an abnormal aggregate of cells. So we give this special name to granulomas in the course of a tuberculosis infection called tubercles. And this is basically, we've got some cells in the center of that tubercle that are still alive and not really metabolizing very quickly or using up a lot of resources. And then there's a bunch, they kind of surround themselves by a bunch of dead cells. And sometimes they'll deposit some collagen here on the outside. This is a structural protein. Okay, and then the organism can kind of hang out like that deep within our lungs for really an extended period of time. We have different names. Sometimes those tubercles can look a little bit different. So we have some different names to describe those tubercles. Sometimes we see a little bit of caseous necrosis. There's actually 7 different kinds of necrosis. I learned that from a med student. Necrosis just means death of tissues. But this caseous necrosis is kind of where the inside of the tubercle gets sort of kind of runny. It's like, caseous means cheese-like. Okay, so sometimes that happens. Sometimes you get a little bit of a calcification around that tubercle, and then we refer to it as a Gone complex. Okay, so that's a little bit more calcified tubercle. So regardless of kind of how you describe that, when a patient is in that stage where that's kind of a stalemate between our immune system and the bacteria, the host is not going to be infectious. Okay, and the host has no symptoms. Okay, so that's primary TB. Typically, when somebody has the tubercles, they may not even know it. Okay, and those can be there for, like I said, an extended period of time. At some point later in life, though, those live bacteria in the center can break out. And that typically would happen if a host becomes immunocompromised due to maybe some other infection or maybe a cancer diagnosis, or they just come to a certain age when their immunity wanes enough that can happen. But we think that only happens in about 10% of individuals. So the question is, if somebody has primary tuberculosis, do you bother to treat it? That's a difficult question. You have to look at kind of each patient, how old they are and their health status and all that. But when those tubercles rupture, and now live mycobacterium are spilling, into the bloodstream or into other areas of the body. Okay, we call that secondary TB, or sometimes we call it reactivated TB. Okay, now the patient is infectious and they start having symptoms. Okay, they have a very productive kind of phlegmy cough, sometimes tinged with blood. Their chest is going to hurt. And then later on, as that, keeps progressing, this is a stage, the late symptoms, where it kind of consumes the patient. So they are feverish, chills, night sweats, they have no energy, they have no appetite, they lose weight. The paleness, because they're not getting enough oxygen out to different parts of their body. That's where they have this ghostly like pallor that used to be in fashion actually in the late 1800s. They get tired really easy. That's where the name White Death came from, to describe this infection. And then once the infection leaves the lungs, we consider it third stage tuberculosis, disseminated, or sometimes you hear this term miliary TB. Okay, when it gets to this point, it's going to be 100% fatal without treatment. And really pretty, maybe like 30–40% fatal even with treatment. So this can really impact any part of the body. You lose kidney function, it can get in the bones, it can enter the brain, end up in coma or cardiac arrest. So typically, again, takes a long time to get to that point, and some people won't ever get to that. Okay, so the diagnosis can be complicated, and probably some of you have recently had a TB skin test. So that's typically, if you're going to start working with patients in healthcare setting, you usually have to have a TB test just to make sure that you're not have some stage of the disease. So how that works, they inject this, it's called PPD, purified protein derivative from the Mycobacterium organism. It's injected under the skin, and then you have to come back between 48 and 72 hours later to have that injection site checked. I think they usually do it in your upper arm. Is that where? Yeah, okay. Oh, even or down there. Okay, like in the picture. All right, so you'll have somebody trained to check and see if there is a lesion. And it has to be of kind of a certain size and raised, to be considered a positive test. What does a positive test mean? Well, it means that either you've been vaccinated at some time, it could mean that you have primary TB, or that you have secondary infectious TB. It could mean any of those things. Or it could mean that you were exposed once and fought off the infection and don't even have tuberculosis or anything. So, but it is a good place to start. If you don't have any reaction, then okay, you're good. You don't have TB. If there is a reaction, well, actually, let me mention one other thing. Here, an alternative to that skin test is a TB blood test. And they don't do this as often because it's a little bit more expensive. And in this country, most people have not been vaccinated. So it's not a huge advantage. You don't get too much more information. But this is often also called an interferon gamma release assay. So that they have, they call it the IGRA assay. And if you remember back a few weeks ago, we were learning about type 1 and type 2 interferons as a kind of cytokine that we talked about. The type 1 is alpha and beta that's helpful to kind of slow down a viral infection. Interferon gamma is type 2. And we don't really talk about that, but apparently T cells release interferon gamma when they have been exposed to this organism, Mycobacterium tuberculosis. So they will take some of a patient's blood, mix it with the organism, and if the T cells in the blood recognize it, they'll release interferon gamma. So again, it's a little bit more expensive. It's kind of nice. The patient doesn't have to go back and have their test read and get the results back very quickly. And the big advantage, places that are people commonly get TB vaccines, like in Europe and Africa, that doesn't cause a positive result. Okay, so if a patient has a positive result from either a skin test or a TB blood test, typically the next step is to do a chest X-ray and look for tubercles. where they are in the lungs can maybe give a hint as to whether a patient has just primary TB or activated TB. But primarily the reason they do the chest X-ray is just to rule out that somebody doesn't have active TB. So if the chest X-ray is clear, okay, good, you're on your way. One of my colleagues, Dr. Thomas, was vaccinated as a child against TB, grew up in Wales, and he's had to have numerous chest X-rays just to rule out because he had that vaccine. Okay, if a patient, if tubercles are present, then typically the next step to try to understand if that patient is infectious or not is to do some microscopy of the sputum. It could do an acid fast stain. An even more powerful tool, though, is to do some kind of fluorescence microscopy where you get a fluorescent dye that will recognize the mycolic acid. Sometimes they use antibodies to do that, really specifically antibodies that will detect that mycolic acid. And then, if they are there, it's just really obvious. Only that Mycobacterium tuberculosis will fluoresce. And if you see that, then the patient has active TB, they're infectious, and they definitely need treated. So it's kind of a laborious process to try to understand what stage that a patient might be at. Treatment is also complicated. Of course, that first drug in 1946, streptomycin, was the first one that we had. That was a miracle drug. We talked about some of the problems with streptomycin. Remember, that's an aminoglycoside, high toxicity to the kidneys and to that cranial nerve, number 8, I think it was. So ototoxicity. So it can cause hearing loss if you don't get that dosed out just right, or whatever. It can cause ear problems. So again, that was a good first step. Essentially, this is rarely used today because most strains of Mycobacterium tuberculosis are resistant to it. So luckily, we don't have to rely on that. For just our kind of garden variety tuberculosis that's not considered highly resistant, we almost always use a cocktail of 4 different drugs. I just named 2 of them here because we talked about them before. Remember, isoniazid targets mycolic acid synthesis, and rifampicin is an RNA polymerase inhibitor. A couple of others that we didn't talk about before, so I didn't want to burden you with, but it's usually these 4 different drugs. Patients are usually taking a handful of drugs a number of times a day. And the problem with these drugs, again, you have to be on them at least 6 months. That's a long time, and side effects are really common. Things like nausea. What else? Tiredness, headache, rash. Okay. Patients, maybe when they start feeling better after a month or 2, might say, I'm tired of feeling nauseous all the time. I'm not going to take those antibiotics anymore. And then it comes back because it, again, takes these organisms a long time to be killed. So you can see why they have this direct observational therapy where nurses will come and watch patients take their medications, a number of times a day just to make sure that they're keeping with them. So that's just for garden variety, again, non-resistant TB. The problem is there's many resistant strains showing up. Multiple drug resistant tuberculosis, this is about 10% of new cases, are resistant to those first line, those 4 drugs, isoniazid and rifampicin. So we have to come up with other things that maybe have even worse side effects, and maybe we have to take them for 9 months or 18 months. It's a long process. About 1 to 3% of new tuberculosis diagnoses are of multiple or extensively drug resistant, or XDR strains. So that means we're pretty much running out of things that might help. They might throw streptomycin. Well, I mentioned that already before. We do have one newer drug, bedaquiline. It was new in 2012. That was the first new drug with a new mechanism to treat tuberculosis, in 50 years. It interferes with ATP synthase for the mycobacterium. I don't know how it targets that, the bacteria's and not ours, but that does help. At least we have one other drug that we would save for those cases that nothing else has worked. Okay, so it's really a trial to treat it. What about a vaccine? Seems like it might be better just to prevent people from getting it. We do have a vaccine. It's not a great one. It works pretty well in kids, but not so well in adults. This came out in 1921, right? So the vaccine that we're using right now is over 100 years old. We haven't improved on it. So there's been a big push by the WHO, the World Health Organization, to throw some more money at this. But it seems like we should be able to get a better vaccine. It's called the BCG vaccine. That stands for Bacille Calmette Guerin. Okay, two French guys that developed it. I think Bacille is just French for bacillus. It is an attenuated vaccine. So it's derived from Mycobacterium bovis. And then it has been cultured over and over and over and over in cells so that it's lost some of those virulence factors. So without even totally, understand how it works. What else was I going to say about that? I forget. Oh, the other problem is, although we have Mycobacterium bovis in cattle, we don't have a good model for Mycobacterium tuberculosis because it's only a human disease. So we don't have a good animal model. And apparently that has hampered the efforts to make a vaccine. Like I said, it's pretty effective in kids, up to 80%, lasts for maybe 5 to 15 years, but it just doesn't work as well in adults. Okay. Although if adults do contract the disease after they've been vaccinated, it's typically less serious. And of course, it's going to complicate the diagnosis process because they will test positive with that skin test. Okay, so let's see, questions about TB? Yeah, kind of a complicated thing that's not going away anytime soon. Okay. All right, well, we'll move on into some gram negatives then. So gram-negative cocci. If you're in lab, you may have noticed that we have not worked with any gram-negative cocci. We've worked with gram-positive cocci, some gram-negative rods, some gram-positive rods too, but we haven't grown any gram-negative cocci. The reason is the really famous ones are quite nasty pathogens in the Neisseria that we're going to talk about here. And they're quite difficult to grow in lab. We call them fastidious, and that means that they have some really kind of specialized growth requirements. They need a particular kind of media to grow on. I think it's called chocolate agar. It's kind of difficult to prepare. Apparently, the Neisseria, when they get into their stationary phase of growth, if they run out of glucose in that phase, instead of just kind of persisting like a lot of organisms will, they lyse. Okay, so it makes them more difficult to grow in the lab. They typically will present as diplococci. So you can kind of see that here. If you look really close, they tend to grow together in pairs. So they divide and then two of them will stay stuck together. This is supposed to be a gram stain. I don't know if these big blotches are some kind of blood cell or not. It doesn't really look like a gram stain like what we do in lab. I don't know if maybe the safranin they used is a little bit more concentrated, make it look more red rather than kind of pink, but that's supposed to be a gram stain and gram negative. They're non-motile, just like the other cocci that we've talked about, the staph and the strep. Okay, they don't have flagella around them, so they'll just more so flow in the medium rather than kind of move purposefully, and they're aerobic. Okay, so we find them in places of the body where there's access to oxygen. So the two famous organisms that we'll talk about, Neisseria gonorrhoeae and Neisseria meningitidis. As if Neisseria isn't hard enough to spell, try spelling gonorrhoeae. It's one of the toughest ones. This is also, it used to be called gonococcus, and then maybe 40 years ago, we renamed it Neisseria gonorrhoeae. And the Neisseria meningitidis used to be referred to as, I'll see if I can spell this, meningococcus. Okay, so Neisseria gonorrhoeae is the causative agent of gonorrhea, which we'll talk about next. And then Neisseria meningitidis causes what we call a meningococcal meningitis. So we've talked about other organisms that cause meningitis, like strep pneumoniae. There are viral meningitis. This is kind of a particular kind of meningitis. So we'll get to that probably next time. All right, so Neisseria gonorrhoeae. Chapter 22, I believe, is bacterial pathogens of the urogenital system. So find a few questions about that for homework. This is a true pathogen, very low infectious dose. Anybody can get it. How is gonorrhea transmitted? By sexual intercourse. Okay, so it's carried by sperm and vaginal fluid. So differences is what structure is targeted based on sex. So males, the urethra is targeted. And for females, the cervix. Okay, and males are much more likely to give it to females. So an infected male is very likely to give it to a female just because it hitches a ride on the sperm. An infected female will only give it to a male a much lower percentage of time. Okay, the other way it's transmitted is from mother to child. Okay, during birth, and that the way that will manifest in the newborn baby is with this really nasty conjunctivitis in the eyes that they call gonococcal ophthalmia neonatorum, and that can result in blindness. So an infection that can lead to blindness. So for that reason, newborn babies are always treated with some antibiotic drops. Years ago, they used to use silver nitrate. Antibiotics are a little bit less irritating to newborns' eyes. They just treat everybody because a mother may not admit that she may be infected or is infected, or she may not know, because oftentimes there's asymptomatic cases. So they just treat everybody, and just kind of easy way to head off that potential infection. I think we're a couple minutes early here, but I think that I'm gonna let you go a couple minutes early, and we'll hit those virulence factors next time. There's a couple interesting ones that are a little bit complicated. So we'll start out fresh next time. We're doing good for time. Okay, have a good day.